to the Division of Education, Innovation and Energy e-learning program. I am Lenetta Clarke, your biology facilitator for today. Today's topic will be the exciting area of genetics under the heading of continuity and variation. Can you roll your tongue? And by roll, I mean fold like this. Be sure to ask other members of your family at the end of this program if they too can roll their tongue, and you would be able to explain to them why they can or cannot roll their tongue. What do you need to continue with this topic? You should have covered areas of cell structure, mitosis, meiosis, and you should understand terms like chromosomes, DNA, and gene. At the end of today's program, you should be able to define terms associated with genetics, explain inheritance of traits, and predict results of simple genetic crosses. Now, we first have to go back in time, back to the 1800s, to a gentleman called Gregor Mendel. He was a monk, but he was also a botanist. Over the course of 10 years, he focused on trying to understand how genes and traits were inherited. He chose the simple pea plant, and 28,000 plants later, he observed that whether he looked at traits like height, color of seeds, or shape of the seeds, he always got the same ratios in the first and second generations. Now, Mendel did not have data or know things like chromosomes and nucleus of cells, but we know that now. He, however, did make some amazing deductions that we use even to today, and that is why Mendel is called the father of genetics. One fact that Mendel gave us was that traits are inherited in pairs. We know that humans have homologous pairs of chromosomes. So here we have one chromosome, and here we have another chromosome. So these two are considered homologous chromosomes made up of sister chromatids. Similarly, we have one chromosome here and another chromosome here consisting of sister chromatids. Each pair of chromosomes contain genes. Remember, a gene is a segment of DNA that codes for a trait. So, for example, this band here would be genes, and let us say it's the genes for eye color. The gene for length of hair may be there. The gene for shape of your earlobe may be there. So let us now look at some terms that we will have to use in going forward. So our first term is an allele. An allele is an alternative form of a gene. And let me explain. Let us use the example of eye color going forward. And in our society, we have dark brown eye color and we have black eye color, just for example. So the alleles for this gene could be brown or black. So the alleles are alternative forms of a gene, the different version that a gene can exist on the both chromosomes. Let us look now at the terms of homozygous, and heterozygous. Now, in genetics, we use letters to represent different alleles. So we would use the letter B to represent eye color. To represent eye color. Now, because we are using the letter B, we can have capital B and we can have common B. So when something is homozygous, or an allele is homozygous, it contains both capital letters or both common letters. So for example, this is the band for eye color. If the person has two capital B's, it is considered homozygous. The both alleles are the same because homo refers to the same. If, however, we have capital B and common B, then that organism contains heterozygous alleles. The alleles are different. Similarly, we can have common B and common B. In this case, you guessed it, it is also homozygous. The alleles are both the same in this example here. So we have done homozygous versus heterozygous. Let us now look at dominant versus recessive. Dominant refers to that allele that suppresses the expression of the other one. So your dominant allele is usually represented by a capital letter. So in this case, we have a dominant allele here and a dominant allele here. In this example, the second one, we have one dominant allele here and one recessive allele. That's the one in the common letter. In the third example, we have one recessive allele here and one recessive allele there. So we have done dominant versus recessive. And finally, we want to look at genotype versus phenotype. Your genotype is the actual allelic makeup of the chromosomes. And that's very simple. You literally quote what the alleles are. So the genotype for the example on top is capital B, capital B. The genotype for the example in the middle is capital B, common B, what the alleles are made up of. And the genotype for the last example is common B, common B. The phenotype, however, is what is actually expressed. What do you see? What do you observe? So let us assume that in our example of eye color, that black eye color is dominant to brown eye color. And the dominant allele, and we have learned that this is the dominant one, it suppresses the effect of the recessive allele. So we will say, let capital B represent black eye color. So for the example on top, where we have homozygous dominant alleles, this organism or this person will have black eye color. So the phenotype will be black, what we will actually see. In the second example, this person's genotype is capital B, common B, and the capital B, the dominant allele, will suppress the expression of the recessive allele. So this person's eye color will be also black, even though they're heterozygous. But the presence of this dominant allele will suppress any expression of the recessive trait. So we say common B is brown eye color. So in the example at the bottom, we have homozygous recessive alleles, and because we do not have a dominant allele, there is nothing to suppress the expression of the recessive allele. So this person who has two homozygous recessive alleles, they would have brown eyes. Let us look at our first activity. You can either pause the video here and attempt to answer the question on your own, or you can take a picture because you will need this image going forward. So let's look at the question. If a homozygous black-haired capital B dog is crossed with a homozygous white-haired dog, common B, what is the probability that an offspring will have black fur? And I'll be back with you in a minute. Now, if we are doing a written exam, This is the format that you would have to use in answering the question. So let us go. We are going to use the letters B, so capital B and common B, to represent the dominant and recessive alleles for the trait or gene of hair color. Parents' phenotype. Remember, we are crossing a black-haired dog with a white-haired homozygous recessive dog. So the parent's phenotype, phenotype is what you see or what you observe. So it's black hair being crossed with white hair. Parent's genotype. The genotype is the allelic or the genes on the chromosomes. So, lucky for us, this example told us what the genotypes were. So we have capital B, and common B, common B. So this parent here capital B, that's the genotype because it's two different chromosomes being crossed with common B, common B. Okay, so the gametes. Because I know that you have done meiosis already, you know that each gamete that is formed would have half the number of chromosomes. So we have to look at all the possible examples or all the possibilities that each gamete will contain. So in this case, we have gametes with the black-haired dog. All of the gametes will have capital B, and we put a circle to represent the gametes. And for the white-haired dog, all the gametes would have common B alleles. Random fertilization indicates that any gametes can't cross with any other gamete. In other words, any egg can be fertilized by any sperm. And in this case, we have a very easy example because we have capital B and common B. So let us look at the genetic cross here. So random fertilization would indicate that this gamete is fusing with this gamete. And what would be your F1 genotype? F1 indicates first familial generation or first filial generation. So the genotypes, what the alleles of the first generation would be capital B from this parent and common B from that parent. F1 phenotype. Remember, the phenotype is what you see or what you observe. So in this case, we have a capital B and a common B. We have a dominant allele here and a recessive allele here. And because the dominant allele is going to suppress the effect of the recessive allele, we are going to have what we will see would be a black-haired offspring. So in this example, all the offsprings would have black hair, even though one parent has white hair. So we are seeing that the white hair gene has seemed to disappear from that first generation. So what's the question? What's the probability that an offspring will have black fur? And your answer here will have to be 100%. All of the offspring will have black hair. If you got that answer correct, congratulations. Let's move on to the second example. Activity 2. A chemical called PTC tastes bitter to some people, while other people cannot taste it at all. Tasting PTC is a dominant trait. Using the letter capital T to represent the dominant allele and the letter common T to represent the recessive allele for not being able to taste PTC, predict the F1 generation between two parents who are both heterozygous for the trait. Again, you can pause the video now and attempt the question, or you can take a picture with your smartphone and we'll be back to you in a minute. So welcome back, and let's just quickly recap what we're looking at. So we're looking at a chemical called PTC, and the traits are being able to taste or not being able to taste this chemical. So we are going to use, they told us to use the letter capital T and common T to represent the dominant and recessive alleles respectively. So let's go. Remember, we have to use this format if we are doing a written example. So we'd use the letters capital T and common T to represent the recessive and the dominant alleles for the trait tasting PTC. So the parents' phenotype. What did they tell us? They told us that both parents are heterozygous for the trait. So it means that if they're heterozygous, that they contain both capital or dominant and recessive alleles on the chromosomes. So the genotype here, we know that they are capital T, common T, being crossed with capital T and common T. So it means that the parents' phenotype, what you will see is that they are both tasters. How do I know that? Because the dominant allele is repressing or suppressing the effect of the recessive allele. So both parents are tasters. Okay, so what are the gametes? All the possible gametes of this parent will be, some gametes will have capital T, some gametes will have common T. For this parent, some gametes will have capital T, we encircle it, and some gametes will have common T alleles. Okay, so let's look at all the possible combinations of all the different gametes crossing together. So we'll take this first gamete here, capital T, crossing with the first gamete for the other parent. So this offspring or these offsprings will have a genotype of capital T, capital T. Let's look at this first gamete crossing with the second gamete for the other parent. So these offsprings will have capital T, common T, genotype. Now, this other combination, common T with capital T. Now, we normally write the capital letter first, so it's capital T, common T. There's nothing wrong with writing common T first, but it's just how we usually do it. And then we look at the final gamete being crossed with the final gamete of the second parent, and these offsprings here will have common T, common T. So that is the F1 generation for the cross between two heterozygous parents. And what's the F1 phenotype? Remember, the phenotype is what you will see or observe. So in this case, we have two dominant or homozygous dominant alleles. So this parent, this offspring will be a taster, because we established that we'd use capital T to represent the dominant trait. This offspring here is heterozygous. It has a dominant allele and a recessive allele. The dominant allele will again repress or suppress the effect of the recessive allele. So this child will also be a taster. Similarly, another heterozygous offspring, so this one will be a taster. The final offspring will have common T alleles. There are no dominant alleles to suppress the effect of the common T. So these offsprings, any offspring that has common T, or what we say homozygous recessive, will be a non-taster. So we have established the cross, and we want to look at the question. The question asks, predict the F1 generation between two parents who are both heterozygous for the trait. So your answer will be, we'll have a 3:1 ratio, meaning 3 tasters to 1 non-taster. So we have answered activity number 2. Once again, if you got that correct, congratulations. So now let's look at another example. Again, let me remind you, you can pause the video at this time, or you can take a screenshot and attempt the question on your own and then come back in a minute to see if you got the correct answer. So let's go. Tongue rolling is a dominant trait. You can roll your tongue. You marry someone who is not a tongue roller. What genotype must you have that guarantees all your offspring are tongue rollers just like you? Let us use the letter D to represent tongue rolling gene. So we'd use capital D to represent the dominant and common D to represent the recessive alleles for the gene tongue rolling. A quick hint, whatever letter that you use, try not to use letters that have the same shape for the capital and common versions. So for example, W, X, S, and P will not be good letters to use because after a while they tend to look similar if you're writing it over and over. So use letters like D and A that are very different in the capital and common versions. So let's continue. The question says, you are a tongue roller, but you marry someone who is not a tongue roller. So the phenotypes have been spelled out for you. So we'll put you on this side. You are a roller, and your spouse is a non-roller. Parents' genotype. Now, this is where you have to do a bit of working backwards. We know for a fact that because your spouse is a non-roller, and the only way that he can be a non-roller is if he has homozygous recessive alleles, because there are no dominant alleles to suppress the tongue-rolling gene. So, the genotype of your spouse would have to be common D, common D. You, however, because you're a roller, you must have at least one dominant allele. So it's at least capital D. But is it capital D, common D, or is it capital D, capital D? Let us work backwards to see what your genotype will have to be if all of your offspring will have to be rollers as well. So let us look at the possibility of this one. So the gametes here will be capital D and common D, crossing with all common D gametes there. So random fertilization will dictate that this gamete will possibly cross with that gamete. So we will have capital D, common D, genotypes, and this gamete crossing with the other one. Okay, so what would be the phenotypes here if your gametes were capital D, common D? In this case, we'd have a roller and a non-roller, like the other parent. And that does not answer the question. So it means that your genotype cannot be capital D, common D. So let's backtrack. What if your gametes were both homozygous dominant? So it will be capital D, capital D. In this case, we would have capital D, common D for the genotype, and of course it will be the same thing if you cross that capital D with that common D. So the phenotype for this generation will all be rollers. And just to ensure that you all understand, the reason why this heterozygous case is a roller because of the presence of the dominant allele here. So in this case, 100% of the offspring will be rollers. So it means that your genotype will have to be capital D, common D. Even if you marry a non-roller, every one of your offspring will have or will be a roller. Final activity. 2 tall pea plants were labeled A and B. The stamens were removed from all the flowers on both plants and the stigmas were dusted with pollen from a short plant. Plant A produced 72 seeds and plant B produced 66 seeds. All the seeds germinated and grew to maturity. The 66 seeds from plant B all grew into tall plants. Some of the seeds from plant A grew into tall plants and some grew into short plants. So, of course, you would do the usual. You can pause the video and attempt to answer the questions here, or you can take a screenshot and we'll be back in a minute. Okay, so let's look at question 1. Using suitable symbols, give the genotypes of the short plant and the tall plants A and B. So we'll just go simple. We're going to use the letter T. So we say capital T to represent tall plant and common T to represent a short plant. So we're looking at the trait of height of plant. Okay, so question 1 asks, give the genotype of the short plants and the 2 tall plants A and B. So the genotype of the short plant will obviously have to be common T, common T. And then we have 2 tall plants, A and B. And what do we know about plants A and B? We know that plant A produced 72 seeds and that some of the seeds from plant A grew into tall plants and some grew into short plants. It means that plant A must be heterozygous. It must have dominant alleles and recessive alleles. So plant A genotype will be capital A, capital T and common T. And then we're going to use plant B. Plant B, we now know, produced 66 seeds, and all the seeds from plant B grew into tall plants. So plant B genotype will be capital T, because plant B had no recessive alleles to pass on to the second generation. Let's look at question 2. How many of plants A progeny would you expect to be short? So we're looking at plants A and they're asking how many would we expect to be short. So we have to do a genetic cross. So let's use the same format. Plant, so we're looking at phenotype. So plant A is tall and was being crossed with a short plant. The genotypes of plant A, we have established that in the previous question. So we know that the genotype of plant A is capital T, common T, while the genotype of the short plant is common T, common T. Gametes. Plant A can only have capital T gametes and common T gametes, while short plant, all the gametes will be common T. So we can safely write that once. So random fertilization will indicate that any pollen will fertilize any ova. So we have F1 genotype. Possibilities are capital T, common T, or common T, common T. So phenotypes of the F1 generation crossing plant A, which is a tall plant, with the short plant, we have some plants will be tall because of the presence of this dominant capital T here, and some of them will be short. But we have not answered the question as yet. The question asks, how many of plant A would be short? So how many seeds did we have from plant A? Plant A produced 72 seeds, and we are told that some grew into tall plants and some grew into short plants. So what is the ratio here? We have, or the possibility of tall to short, we have 50% of the plants from this course will be tall and 50% will be short. So if we had 72 seeds in total, then it means that half of them will go into short plants and half will go into tall plants. And half of 72 is 36. So approximately 36 of the plants from this course will be tall and 36 of the plants will be short. So your answer here will be 36 plants are likely to be short. So that brings us to the end of part 1 of genetics. We have learned about terminologies like alleles, homozygous versus heterozygous. We did dominant versus recessive, and we looked at simple genetic crosses, and we predicted what likely outcomes will be between crosses and F1 generations. So that brings us to the end of genetics part 1. I thank you for being here with me, and I'm really glad that we were able to achieve all of our objectives for today. So we did define terms. We looked at alleles, homozygous versus heterozygous. We looked at dominant versus recessive. And we did look at phenotype versus genotype. We explained inheritance of traits, and we did some simple genetic crosses and predicted outcomes from these crosses. To end, I'm going to leave you with a series of questions that are past paper style to give you an idea of what to expect in your exams. Please discuss your answers with your local tutors, and we encourage you to join us again for our next segment of e-learning under the Division of Education, Innovation and Energy. Thank you.